Morning. This time I will share drug discovery and development. The topic I will divide into three parts. The first one, overview, drug discovery, and the last is drug development. Now let's proceed to overview. Drug discovery and development is a process which aims at identifying a compound therapeutically useful in curing and treating a disease. The process involves the identification of candidate, synthesis, characterization, validation, optimization, screening, and assay for therapeutic efficacy. Drug discovery and development is lengthy, complex, and costly process. Entrance with high degree of uncertainty, the drug will actually succeed. Inventing and developing a new medicine required 12 to 24 years for a single new medicine, and cost for overall process has escalated sharply up to an estimate 1.4 US dollar billion for a single new drug. Let's see this diagram. Drug discovery and development is a complex process, involve many expertise and stakeholders. Commonly, researchers discover a new drug through new insight into a disease process that allow researchers to design a product to stop or reverse the effects of a disease. Second, many tests of molecular compound to find a possible beneficial effect against any of a large number of diseases. Then existing treatment that had an anticipated effect, and the last new technologies such as those that provide new ways to target medicinal product to specific site within the body or to manipulate genetic material. At this stage, thousands of compounds may be potential candidates for development as a medical treatment. After early testing, however, only a small number of compounds look promising and determined for further study. Now let's move to drug discovery. There are some processes in drug discovery, include target candidate or target identification, target validation, hit identification, hit to lead process, and lead optimization. The first phase in drug discovery known as target identification. This process may be conducted with in silico and in vitro method. This phase is performed to identify target involved in a specific disease. A target is usually a molecule integral to gene regulation and intracellular signaling, such as nucleic acid and or protein. There are several approaches that used to identify the target, such as direct medical method. Biochemical affinity purification provide the most direct approach to finding target protein that binds small molecule of the interest, because they are based on physical characteristics. I mean that because they are based on physical interaction involving mammalian or human protein, biochemical method can lead to information about molecular mechanism of efficacy or toxicity highly relevant to human disease. Then genetic interaction and genomic method. Target identification based on genetic or genomic method differ the relative ease of working with DNA and RNA to perform large-scale modification and measurement. This method often use the principle of genetic interaction, relying on the idea of a genetic modifier, such as enhancer or suppressor, to generate target hypothesis. The last is computational inference method. Computational methods are used in infer protein target of small molecule in addition to providing analytical support for proteomic and genetic technique. This method can also used to find new target for existing drug with the goal of drug repositioning or explaining of a target effect. The A, B, C figure are the illustration of a direct biochemical method, genetic interaction and genomic method, and computational inference method, respectively. Now let's move to target validation in drug discovery. Target validation is the second step in discovering a new drug and can typically take 2 to 6 months. After selecting a potential target, researcher must demonstrate that it is involved in the progression of a given disease and that its activity can be regulated. Conducting careful and precise target validation experiment is essential for the success of drug development in the following stage. There are several aspects of target validation techniques, such as animal model of disease, expression profile, biomarker, cell-based disease model, pharmacological tool compound, functional analysis, in vitro or in vivo genetic manipulation, and disease association genetic. The process involves the application of a range of techniques that aim to demonstrate that drug effect on the target can provide therapeutic benefit with acceptable safety window. Let's see this table. This table provides characteristic of preclinical model for a target validation. Let's see this one: cellular model, animal model, and so on till human model. For this one, we can see that target modulation human are relevant, and causality in human. Let's take out one of these point, for example, human genetic. This one, target modulation, this effective, but with some limitation. And human relevance, highly effective, causality in human, effective, but with some limitation. Now let's move to hit identification. The hit identification phase focus to apply the most appropriate hit finding approach to successfully identify validated hit series with the best chance of developing into that like a component. Hit compound is a primary active compound with non-promiscuous binding behavior exceeding a certain threshold value in a given assay. The active is followed up with an identify and purity evaluation. An authentic sample is then obtained or resynthesized and active confirmed in a multi-point activity determination to establish the validity of the hit. We call this one as a validate hit. Commonly, there are several techniques to finding the hit compound, such as virtual screening, high throughput screening, fragment screening, and knowledge-based design. High throughput screening is a drug discovery process that enables automated testing of a large number of chemical or biological compounds for a specific biological target. See this figure in the right side. Due to utilization of robot detector and software that regulate the whole process, enable a series of analysis of chemical compound to be conducted in a short time, and the affinity of biological structure, which is often related to toxicity, to be defined. Then let's move to virtual screening. Virtual screening can be defined as a set of computational methods that analyze a database or collection of compounds in order to identify potential hit candidate. The aim of virtual screening is to identify molecules of novel chemical structure that bind to the macromolecule target of interest faster and cheaper than in vitro method. In virtual screening method, the discovery of drug is conducted based on protein structure or a ligand structure. Based on ligand and protein structure, there are 4 approaches in virtual screening, such as structure-based design, ligand-based design, library design, and de novo design. The most common method in virtual screening are structure-based design and ligand-based design. One of example, FDA approved drug with design from in silico drug discovery is captopril. Captopril is FDA approved medication used in the management of hypertension, left ventricular dysfunction after myocardial infarction, and diabetic nephropathy. Now let's move to heat to lead. Heat to lead phase is to rapidly assess in a number of heat clusters in order to identify 2 or 3 heat series that have the best potential to develop into the lead. This process starts by confirming that a true structure-activity relation exists within a number of heat series for a biological target. In addition, an early assessment is made of in vitro ADMET properties to help select the most promising lead for optimization. Heat to lead project typically run for 6 to 9 a month. Lead optimization is to deliver one or more clinical candidate for evaluation in the formal regulatory program prior to human clinical trial. Lead optimization aims at enhancing the most promising compound to improve effectiveness, diminish toxicity, or increase absorption. Lead compound is a prototypical chemical structure or series of structures that demonstrate activity and selectivity in a pharmacological and or biochemical relevant screen. This forms the basis for a focused medical chemistry effort for lead optimization and development with the goal of identifying a chemical candidate. A distinct lead series has a unique core structure and the ability to patent it separately. Then let's move to the development. Once researchers identify a promising compound for development, they conduct experiments to gather information on how it is absorbed, distributed, metabolized, excreted, its potential benefit, and mechanism. The mechanism of action. The best dosage, the best way to give a drug such as by mouth or injection, side effect or absorb even that can often be referred to as toxicity. How it affects different groups of people, such by gender, race, or ethnicity differently. How it interacts with other drugs and treatment and its effectiveness as compared with a similar drug. So let's see this one, preclinical trial process, this lead selection of optimization that move to the candidate confirmation, and last is preclinical characterization. We see this one, efficacy assessment. Does it work? ADMET profiling, how can it be delivered? And what does the body do? Toxicology and safety pharmacology assessment, is it safe? And the last, pharmaceutic, is the manufacture valuable and controllable? Then let's move to the difference between pharmacokinetic and pharmacodynamic. Is that pharmacokinetic is defined as the movement of drug through the body, whereas pharmacodynamic is defined as body, as the body's biological response to drug. Pharmacokinetic, pharmacodynamic modeling link those concentration relationship and concentration effect relationship, thereby facilitating the description and prediction of the time course of drug effect resulting from the certain dosing regimen. So let's see once more time. This is pharmacokinetic and pharmacodynamic assessment. Dose of drug administered, drug concentration in systemic circulation, drug concentration at site of action. Then let's move to this one, a distribution. This is elimination. In case of this one, clinical response, toxicity, and effectiveness. Now let's move to pharmacokinetic, ADME tox. ADME is the abbreviation for absorption, distribution, metabolism, and excretion. ADME studies are designed to investigate how a chemical, for example, a drug compound, is processed by a living organism. Toxicology tests are often a part of this process, yielding the acronym ADME. ADME is the process of pharmacokinetic. In drug discovery and development, researchers must examine the activity of drug in the body to assess safety and toxicity. Drug metabolism and pharmacokinetic study, such as ADME and toxicology study, are critical in this process. The data collected tell researchers if a drug is viable and provide specific target for further research and development. Pharmacodynamic is a study of the biomedical and physiological effect of drug, especially pharmaceutical drug. The effect can include those manifest human animal, including a human, microorganism, or combination of organisms, for example, infection. Pharmacodynamic refer to the relationship between drug concentration at the site of action and the resulting effect, including the time course and intensity of therapeutic and adverse effect. The effect of a drug present at the site of action is determined by that drug binding with a receptor. There are several aspects of pharmacodynamics, such as receptor-effector, dose-response curve, agonist, partial agonist, antagonist, inverse agonist, signaling mechanism, and receptor regulation. Then let's move to clinical trial. Clinical trial are a type of research that study new tests and treatment and evaluate their effect on human health outcome. People volunteer to take part in clinical trial to test medical intervention, including drug, cell, and other biological product, surgical procedure, radiological procedure, device, behavioral treatment, and preventive care. This figure, let's see this one, show the step from preclinical trial until a clinical trial. Let's see this one, from this one of preclinical trial, from this one, Then this part is a clinical trial. We start from this one and last to this point. There are 4 phases of biomedical clinical trial. Phase 1, study usually tests new drug for the first time in a small group of people to evaluate a safe dose range and identify side effects. Then phase 2, studies test treatment that have been found to be safe in phase 1, but now need a large group of human subjects to monitor for any adverse effect. Phase 3, studies are conducted on large population and in different regions and countries and are often the step right before a new treatment is approved. Phase 4, study takes place after country approval and there is a need for further testing in a wide population over a longer time frame. Phase 1, test for safety. See this one? A small number of people, sometimes healthy and sometimes with a medical condition, are given a tiny dose of drug under careful supervision, not to test if the drug works, but in order to check for any side effect. Phase 2, sees the drug given to people who have medical condition to see if it does indeed help them. Phase 3, trial are only for medicine or device that have already passed the first 2 steps and involve them being compared to existing treatment or a placebo. The trial often lasts a year or more, involving several thousand patients. There are several regulations in drug discovery and development, such as GLP, good laboratory practice, GCP, good clinical practice, and GMP, good manufacturing practice. These regulations are required in preclinical development, clinical trial, and manufacturing, respectively. Pharmaceutical regulation across the world play an important role in ensuring the safety and efficacy of the approved drug. After a registration proceed and review by FDA, if the drug was approved, the pharmaceutical manufacturer will get an exclusivity right for the newly the product for several years. Post-market surveillance refer to the requirement that manufacturer monitor their medical device or product after being approved for sale and seeing in market use. This monitoring conduct every 6 months for 2 years. So let's see this one, the last part of our discussion for this morning. Let's repeat one more time the registration and post-market safety. Let's see this one. This is a clinical trial, preclinical, phase 1, phase 2, phase 3, FDA review and approval. Then we have phase for drug approval. Then that's all for this time. Thank you so much. And again, take care. Thank you.